coming down the pipe for 48 hours or so. So let's go ahead and take a look at where we left off. So we're talking about sarcomeres. Now, sarcomeres, remember, are basically, these are your contractile unit. This is your fundamental unit of the muscle. This is your contractile unit. And when you take a look at your sarcomeres, remember what's in the sarcomeres is all those myofilaments, right? So we talked a little bit about the actin filament and the myosin filament, and there's a bunch of other proteins in there as well. But because of the nature of these filaments, it's what creates the striation patterns associated with skeletal muscle cells. So those striation patterns are actually because of the organization of the sarcomere. So this is basically taking a look at the organization of the sarcomere. We start first with what's called the Z disc or the Z line. And basically what the Z line is like a network of protein. It's kind of like a wall. That defines the boundaries of a sarcomere. So for instance, one sarcomere to the next is defined by one Z line to the next. So basically you take the Z line, which kind of has a little zigzag to it. And so this Z line, basically, this distance will be one sarcomere. And of course you have another Z line over here. So this will be your second sarcomere in line and so forth. So the distance between two Z lines is going to be one sarcomere. This also basically is an anchoring point. This is the reason why I like to think of it as sort of like a wall, because it's like a wall that you tethered yourself to. And in this case, what you're doing is you're tethering your actin myofilaments to it. Okay? Now, in the sarcomere, you'll actually see the Z discs, and they kind of create a dark area of the sarcomere. Now, there's other types of lines and bands and things like that in the sarcomere. And so what I want to do is sort of introduce the lines and the bands. And we're going to see a picture of the sarcomere, and it'll be a little bit more obvious there once we actually see the picture of it. So within the actual sarcomere itself, we have several different regions. The first one is what's called the I band. So the I band basically is an area of light stain. It'll have a Z disc in it typically. And generally what happens is it goes from myosin filament to myosin filament. So if you take a look at an I band basically will encompass a Z line like this, okay? And remember, it's fairly light staining. Now, it's a little, the accuracy of the spacing on that is not quite as good. I'm still, after all these years, not really great at drawing sarcomeres. So the pictures are actually much better usually than what I can draw. The next big band that we see is what's called the A band. And the A band is important because now this is a band that is a dark staining area. And it happens to be an area of dark stain. Why? Because you have an area of overlap. So this is an area of overlap where you see the overlap of actin and myosin. And generally speaking, it's in between I bands. So the A band is kind of like right here in the middle. And it's kind of darkly staining. Then you have what's called the H zone. This is an area in the A band where there's a temporary little area of non-overlap. And so it's a little bit lighter. So there's a little bit of a lighter area there. And right down the middle of the H zone is a line called the M line. And this is basically protein filaments similar to the Z line, only a little bit smaller. So this is kind of like a thin wall. Attaching the myosin filaments. So basically what you have is an M line to which you have myosin attached. Then you're going to have a zone of overlap between your actin and a zone of no overlap. But the actin itself is going to be attached to the Z line, like that. So your A band is going to be this zone of overlap. The H is going to be right here. And this guy right here is M. The I band, if you're wondering, is going from non-overlap region to non-overlap region. So that's your I. So I always have to build the sarcomere backwards because it doesn't make any sense to me unless I build it backwards. So there's a couple of other things, however, when you see this, right? So basically your I bands and your A bands, this is essentially where you have your myofibrils kind of aligned next to each other in this sort of alternating dark, light staining area. That's what gives you your striations, that kind of light, dark, light, dark, light, dark that you see in skeletal muscle. That's what that is causing, is the alternation between the A bands, the darkly staining A bands, and the lightly staining I bands. There's also another protein filament called titin. So titin basically is a long chain of amino acids. And so this kind of helps to sort of make the muscles a little elastic. So think of it as sort of like a bungee cord. That's kind of tethering everything together. So it kind of keeps everything elastic and extensible. We'll take a look at the role of the titin filaments here in just a second. They do play a role, but we have to understand the sarcomeric structure a little bit better first. So here is our sarcomere. So here you can see your Z lines right there. So those are Z disc or Z lines. So that's basically bounding one sarcomere. So between two Z lines is one sarcomere itself. And then you can see this alternating kind of line, right? So this kind of light colored line right there, that's the I band, right? Notice the I band is basically a region of actin only. And it's tethered to the Z line, okay? That's the reason why it's light, because you don't have as much protein filament there, right? And not only that, but it's the thin filament. So it's even less protein material there to actually stain. That's the reason why it's light in nature. Wait a minute. So you're prattling on. Good. Okay. For a minute there, I had flashbacks. I was like, no, I have to be recording this. I'm not doing this again. Okay. Then as you start to move in, you can see this darkly staining A band, where this is the region of overlap. And that region of overlap becomes very important. Until you get to the I band, which is this little clearing area right here, right? That's the H zone right there. That's the little open area right there. And you can see this like little dark shaft going down the middle. That's the M line. Okay, so those are all the different functional pieces. And this is actually what it looks like. So here you can see your Z line here. You can see your actin filament attached to the Z line. And then in between, you see the myosin filaments in between the actin attached in the center by this M line. So you kind of have this zone of overlap, right? And notice what the myosin looks like. The myosin basically is a motor protein with kind of like a shaft looking section, a filamentous section, and then it's got a head section. So the head section is the part that actually moves. So myosin kind of looks like basically your wrist, and your arm is like the shaft portion, and then basically you can pivot the head. So if I want to grab onto something and pull it, I can just basically do a little wrist action and grab it, pull it, grab it, pull it, grab it, pull it, grab it, pull it, right? So that's kind of what a myosin is doing. And so it's a type of motor protein. So you need ATP in order to pull this off. And so that's basically what your myosin is. Notice you don't just have one. You basically have a whole bunch of them all bundled together. So you can see like all these little itty bitty myosin heads in there, these little golf clubs, and they're all attached to the actin. So when you take a look at this, you can see these myosin heads, several of them in series, and they're basically attached to your actin thin filament, right? And so you don't just have one myosin head pulling on the actin, you've got lots of myosin heads pulling on the actin. Each one of those myosin heads are requiring ATP in order to actually get that swiveling pivot action that you need in order to actually pull that actin to the side. So ultimately then, what happens is when you take a look at your thick filament, you can see this is a kind of a close-up version of your twisted golf club. Right, that's kind of what your myosin looks like. But when you take a look at this, this is also going to show you a little bit about what titin looks like. So see this little corkscrew looking guy right there? I can't really drop because it's too blown up. But these little corkscrew looking guys right here, this is titin. And so what titin does basically is it anchors the Z line, but then it actually kind of grabs onto the myosin filaments and sort of tethers this entire myosin structure from one Z line to the next. Think about it for a second. If all you have is non-overlap in your H zone, where all you have is just these shafts of proteins, that would look like a point of weakness, wouldn't it? Because you have nothing there to support it. It's just simply protein. So if you put any kind of stress on that, like lateral stress on that, you're going to bend it and distort it, and that's going to compromise the structure of the sarcomere. So in order to shore that up, so it's not so noodly in the middle, that's a technical term, by the way, right? Noodly in the middle, what you do is you basically anchor it to your structural support, right? Your Z line is your structural wall, and use titin to basically anchor into the structural support. And then you basically connect titin to titin, Z line to Z line. And notice how you have a continuous thread of titin that shores up on both sides these myosin filaments. So what that does is it kind of creates a solid substrate that will help support this vulnerable myosin in the middle, right? Now, granted, yes, you do have the M line. That certainly does shore that up as well. But titin is kind of like a little extra veneer of stability for your myosin filaments. And so this is basically what the structure of a sarcomere looks like. And when you take a look at it end on, which is really helpful, it kind of helps give you a really clear view of what these filaments look like in 3 dimensions, right? So first of all, when you take a look and just peel away everything except for the actin, you can see how you're looking at the ends of these actin filaments, right? So in this particular situation, what you're seeing is just what the I band looks like, because the I band is just this region of non-overlap where you just have actin tethered to the Z lines, right? So it's just actin by itself. There's no myosin in there. And so all you see are these little filamentous rods of actin. And none of that, but I want to draw your attention to the way they're arranged. Right, so these actin filaments are arranged in all 3 dimensions, right, in a circle. Oh, excellent. Yeah. Benson just walked in with the muscle fiber. Yeah. Yeah, because we're just about there. Yeah, right. Incredibly appropriate for where we are. Right, so you notice how they're basically arranged in 3 dimensions, so that if you're in the middle of an actin, you've got 360-degree contact to your actin filament itself. right? Now, when you move into the A band, remember this, the A band is the darkly staining region, right? This region of overlap, this area right up here. Now what you have is the overlap of your actin with your myosin in between it, right? So you have your myosin, they're all overlapping in this particular case. And so what you see is the combination of both. So notice you see the actin arranged in this hexagonal shape, but in the center of it, you see the myosins also contacting every single corner of the hexagon. And in every single corner of the hexagon, you're going to find a filament of actin. Now, what are the little blobby stars on the myosin? Those are the heads. Each one of these little blobs on the star is basically the myosin head. The central portion is the filament, the twisted golf club handles that form the filamentous core of the myosin. So what you have then is not only do you have multiple heads in contact with your actin filament, but you have multiple heads of myosin in all three dimensions wrapped around this actin. So you have myosin all around pulling on this in all three dimensions. Okay, that gives you a lot of power. Now let's go ahead and move into the H band. Remember, the H band is an area where your actin stops, right? So if you take a look at the H band, remember, your actin stops in the H band, right? It cuts short, but your myosin continues. So now what you should see in the H band is only your myosin, absent the actin. And that's exactly what you see, right? So this is your zone of non-overlap, where you only have your myosin and not your actin. And then finally, the M band, this is cut right down the middle of the M line, this big chain. You take a look at the M line, which is right here, you should basically see a protein wall, which is what you see. So this is like your protein matrix here. And then within that, you should see the normal organization of all of your myosin, right? Which is what you see because your myosin is connected to the M line. Okay. So I love these images because it kind of cuts the sarcomere in cross section and allows you to kind of be able to see how you change in state from one section of the sarcomere to the next. So you can kind of get a clearer and better idea of how this is looking and how this is working. So let's take a closer look at the thin filament. You guys are like, well, wait, didn't we just see the thin filament? No, actually, we just got started on the thin filament. So the thin filament is certainly made of actin. Yes, that's the easy part. But there's more than one type of actin in the thin filament. There's two types of actin. There's one that's called the fiber, so the F actin. So this is basically the one that is largely making the long linear fiber structure of the actin filament. So it's kind of like the structural body of the actin itself. But there's another type of actin called G-actin. And the important thing about G-actin is in G-actin, the G-actin has an important active site. And this active site is basically for myosin head binding. Which makes a lot of sense, right? I mean, if you're a myosin head and your job is to grab onto the actin and pull on it, then the first step is to actually allow you the necessary place to bind to the actin in the first place, right? You got to have some place to grab, and that's what G-actin allows you. And this is a necessary critical sort of a thing. However, there's more to this actin thin filament than just the actin. Also wound into this structure is another protein. So you have two proteins here, F-actin, G-actin, and then you have tropomyosin. Tropomyosin is a long kind of a rope-like molecule. And its job is to cover the active sites of G-actin. And when it does so, it blocks the myosin head binding. Important, right? Because the idea here is you don't just want a muscle that's contracting willy-nilly. You want the muscle to largely be off and to only contract when you tell it to contract. So the only way that you can tell it to be off is by blocking all those sites that would activate it. Right? So tropomyosin's job is to basically block this active site. Okay, there's obviously a problem now, right? You've got it blocked, great. But what if you want to contract? Don't you need access to that site in order to contract? The answer is yes. So there's another protein involved here, troponin. And there's different forms of troponin. There's a cardiac troponin, which is actually a common marker for heart attack or some sort of a heart dysfunction. Matter of fact, it's a metric that a lot of times doctors see that. They will put you on the red flag list because it only gets shed when the heart muscle gets damaged. And so it's a clear marker for bad things are occurring to you. Actually, there was I remember one patient that we had who was getting ready to do a stress test, an echocardiogram stress test, which is basically kind of like a nuclear stress test, only they do an echo instead. And they kind of run on the treadmill for a little while, and then they lay down, have the little ultrasound done of their heart to see kind of how their heart's beating and all the flow dynamics that are going through the heart so they can measure everything. But just on a whim, our cardiologist decided to check his troponin levels really quickly. And as it turned out, his C troponin levels were really high. And the doctor pretty much pulled the plug on the test. Like, no, do not get him on that treadmill because he will not come back off of that thing alive if you put him on that treadmill. So luckily he was, and that doctor that was his normal. A lot of doctors are really good at what they do, but I mean, I've never actually seen a doctor pretty much avert disaster as many times as he did and not be like an ER doctor or something like that. Dr. Richard Jantz, I think he's retired now, but one of the all-time greatest doctors I've ever seen in action. Great guy too. If you want to be a doctor, get to know him. He's the kind of doctor you want to be. Now, I would ask you, why do you want to be a doctor? But that's just me. But he was very, he's still very much, and he's been in his 50s when I worked for him. He was still very much driven by that sort of like, I'm here for people. I'm here to save people. This is why I'm in cardiology. I mean, he flew to Sydney, Nebraska, like twice a month, just so the people in Sydney, Nebraska could have a cardiology presence there. I mean, he pretty much went everywhere. I mean, it's like, have heart, I'll doctor you. Just like, it's like he had a cardboard sign that said, "Let me save your life." Right. I mean, it was an unusual sort of a thing. He was very driven, very quiet guy, very smart, very driven that way. So don't really meet very many of them. Most doctors are usually on the hard edge side, usually a little too much stuff. There's another cardiologist that works things like that, but she's also very good, just not very warm and cuddly. But him, he was just like, you could just sit next to him drinking beers and it would just feel normal. It's like, it's like, okay, yeah, we're not a doctor now, we're just. So basically, what is troponin? So troponin is a protein with 3 different domains to it. And so when you take a look at the troponin, it does 3 things. First of all, one of them binds to actin, which makes a lot of sense, right? I mean, after all, you're part of the thin filament. So probably if most of your thin filament structure is fibrous actin, then probably you'd better bind to actin. I mean, how else are you going to be associated with the filament, right? So that one makes sense. So that's a big one. The other one is it also binds to tropomyosin. Remember, this is the rope that's covering up your active site in G-actin. And the third and potentially the most important one is it has a binding site for calcium. So basically what this does is it binds to all three, but not all at the same time. Right. Now, as is the case often with proteins, whenever you bind to something new, like say, for instance, calcium, it oftentimes precipitates a shape change, and that shape change has beneficial consequences to it. So the way that you regulate access to your active sites on G-actin is with this troponin tropomyosin complex. So the tropomyosin is there to cover up the access. The troponin is there to give you access to those sites at only certain times, right? So only the times when you need to actually contract. And we'll take a look at an image that should help to kind of clarify a little bit of everything that's involved in that thin filament piece. Okay, now what about the thick filament? Is it as convoluted and multivariate as the thin filament? And the answer is no. Right. I mean, the myosin thick filament kind of mostly is what it is. I mean, largely what you have is a heavy chain and you've got light chains. So basically, when you take a look at heavy chains and light chains, all you're really doing is essentially building the long central shaft, right? So the heavy chains are forming the shaft. So these are like the shaft protein, the shaft part. And then your light chains are basically building the rest of it, right? And so the myosin heads themselves will then kind of extend out, and those will be part of the light chain itself. Now, the important thing to remember about the myosin heads is that when you build your shaft, your head is actually not connected to the shaft. It's actually connected to the light chain, which kind of has a little bit of a wiggle to it. So that allows you to be able to distort that light chain and swivel the head, right? It's just like my hand is not connected to my arm, right? There is a lot of swivel in there in the carpals that allows me to be able to pivot my wrist. Okay, it's the same basic idea. If it were attached to my radius and ulna, I would not be able to move my hand like that at all, right? That'd be kind of weird. But that's kind of like the way cats and dogs are, right? I mean, they don't have a whole lot of carpal movement. I mean, they do have a little bit, right? But not like us. They don't have as much fluidity of movement. And so, like, especially as other animals are just like, I mean, all they have is like a solid arm and they just kind of like that, right? I mean, that's kind of what they do. So it's just kind of more like a big foot, right? Minus the ankle. Okay, so let's see what the myosin heads do. Obviously, the first thing they do is they bind to the active sites in your G-actin. When they do this, they create what's called a cross bridge. This is basically a myosin head connected to the active site on G-actin. So then what you have is then the myosin head attached to the actin. And then, of course, you have this hinge region that connects the head to the shaft, right? And then you basically have to have ATP consumption. In order to have that, you have to have ATPase, right? This is an enzyme that basically breaks down ATP. And what it does is it breaks ATP into ADP plus phosphate plus energy. And of course, you use the energy to pivot. All right, so that's what you use the energy for. So muscle contraction requires energy, right? Which is the reason why we all eat and eat and eat and eat and eat and eat. How do I know that? Don't make me ask how many times you've eaten today, or yesterday, or tomorrow, or the day after that, right? Unless you're fasting, that doesn't count, right? Most of us are eating every day. Wow, that's a lot of food, right? So that's the reason why part of that, obviously, is because you need ATP in order to motivate muscle contraction, for instance, and of course, a whole host of other things. Anything that you want to do work-wise, biology, like on a cellular level, on a physiological level, on a tissue level, on an organ level, you need ATP to do it, right? So this is kind of what the thin filament looks like. So let's take a look at it. So here you can kind of see the nice image here. So here you can see this is going to be your F actin here, right? So these guys right here, these kind of basic ones. So these guys are the F actins here. And then the G actins are going to be the active sites, right? So that's going to be these guys here. And so these are your active sites. So ultimately what's going to happen is your myosin head is going to bind to that area, to that little G site. However, the problem is these sites are covered up by tropomyosin, which is kind of like the big, long, ropey area. Right now, it looks like it's pulled off, but normally tropomyosin, which is kind of like this long, twisted rope, so you can actually see it is a rope-like molecule. It looks like a noose, actually, right? But that somebody just kind of cut the loop on, right? So basically, normally what happens is this is a little bit more slackened up, and the myosin actually, the tropomyosin actually kind of drops down here and actually covers up these areas. So as this kind of like comes down, it actually covers up these active sites like this. So that's usually how the tropomyosin works. This one's been tightened up a tad. So this is your troponin. Notice you have here the connection to your tropomyosin. So this is your tropomyosin binding area. This one up here is going to be your actin binding area. And this one here is your calcium binding area. So ultimately then, this is your entire filament. So basically, how do you actually manage to gain access to all of this? Well, the way that you gain access is simple. What you do is you basically take calcium, and then normally at rest, what's happening is the tropomyosin rope is covering up the active site, so you don't have access to it. However, and troponin is attached to the tropomyosin, so it's just kind of hanging out there. And it's kind of got one hand on the actin as well, right? So it's kind of hanging on like this. So then what happens is calcium comes in, binds to the calcium domain of troponin. When it does that, it changes the shape of troponin. So troponin changes its shape. And as it does so, it basically grabs and sort of pulls the troponin rope, like you're rolling over on the covers over yourself. And as it does that, it pulls the tropomyosin rope off of the active sites, leaving them available now to bind to the myosin heads to form that cross bridge. Once you form the cross bridge, then all you have to do is pull. We'll talk about that in a second. It's called the sliding filament theory. And we'll talk a little bit about that in more detail. However, before we get there, right, so we talked about a little bit of stuff. We talked a little bit about structure. We talked about the structure of the sarcomere, the contractile unit, right? We talked obviously about the muscular structure, right? The entire muscle fiber development and the organization of it. But now what we want to do, so we've kind of been talking about like lots of little stuff, right? The micro stuff, like how everything deep inside is sort of arranged. And now what we want to do is we want to go back out. And what's happening here, if it seems a little chaotic, it's because it's intentional. Because what we're doing right now is we're taking kind of a modular approach. So we're basically taking a particular module in a very long, windy process. And we're sort of unpacking each individual module. And then we're moving on to another module, unpacking that one. And once we unpack all the modules, we're going to tie them all together in their linear, continuous process. Okay. So we talked a lot about the muscle, the fiber arrangement, the fascicular arrangement, the sarcomeric arrangement, the arrangement of the major fibers and their structure. So we got a lot of microstructural detail now. Now what we want to do is we want to sort of back out, move back up to the beginning of the process to before we get to the muscle, right? So it's great that you have a muscle. It's great that you have a sarcomere that contract the muscle. But we still have a couple of more things to talk about sarcomere. But what we want to do is we want to ask this question. How do you make it all happen in the first place? Right? Who tells the muscle what to do? Well, that's where we get to this one, the neuromuscular junction, or the NMJ. So the neuromuscular junction is basically a synapse. And in every synapse, there is a common structure, right? There's basically a presynaptic cell, typically the neuron. Then there's a synapse itself, and the synapse itself is basically this little space in between the presynaptic cell. So this would be presynaptic. And then the postsynaptic cell. Now for us, the presynaptic cell is the motor neuron. The postsynaptic cell is the muscle fiber. Right, so we already talked about the muscle fiber and kind of moved down from there. So now what we want to do is we want to talk a little bit about this connection. And of course, you have a synaptic cleft, the space, the synapse between presynaptic and postsynaptic target. So let's take a look at this, right? So first of all, there's a problem here, isn't there? How do we get to the synaptic target if there's a gap there? Right, because basically when you're taking a look at the neuron, in the axon, in the terminus of the axon, you basically have the need to sort of transmit this sort of neural impulse to your target cell. Right? That's basically what you do. But here's the problem. You've got a gap. So how do you jump the gap? That's problematic, right? Well, as it turns out, what happens is in this the presynaptic cell hanging out there are synaptic vesicles. A lot like just secretory vesicles that you would see just in a general cell. Only instead of releasing to the outside world, they're just hanging out. And what are they full of? Acetylcholine. Now, acetylcholine is basically the neurotransmitter that is associated with motor neurons. And they typically will communicate through ligand-gated ion channels to a muscle fiber. Wow, okay. Let's unpack that one a little bit. We're going to need to obviously go over that one a wee tad, right? So this is kind of like, by the way, this is just a summary slide. This is just kind of the full wrap up of the thick filament and the thin filament and the sarcomere. So that's basically what this one is. We already saw this one. We just saw bigger versions of it. So I just put this one in here in case you guys want to sort of see it all together, because it actually is helpful to see it all together, right? Because what do you see up here? Now, notice what I didn't do before is I didn't talk about any of the stuff up here yet, did I? Because that's where we are now. So ultimately what you have is you have your presynaptic cell, your neuron, your motor neuron. At the motor terminus, basically what's going to happen is you essentially have this nervous impulse that's basically radiating through the membrane of the axon. And then when this nervous impulse, this action potential comes into this area, it's going to trigger the release of calcium from these voltage-gated calcium channels. This is going to release calcium into the neural synapse, the synaptic area, the presynaptic terminus of the cell. That calcium is then going to stimulate these little vesicles full of acetylcholine to merge with the membrane and release the acetylcholine. So these little yellow guys right here, this is acetylcholine. So the acetylcholine will then grab onto these ligand-gated sodium channels, which will then allow sodium to flood inward, and that will reignite the action potential in the sarcolemma. Right, so notice you have ligand-gated channels, which then give way to voltage-gated channels. And then what's going to happen? This action nerve impulse is going to radiate down through the sarcolemma and then go down to the T-tubule. Right, that's where that muscle component came in. Okay, so this is a T-tubule here. So this is the piece where we're at now, is this upper piece, this neuromuscular junction component, right? So whenever you're basically taking a look and you're asking yourself, well, how does the muscle know to contract? Well, it knows because there's a neuron that tells it to do so. How does it tell it to do so? By basically transmitting that action potential, which is just basically like a rolling voltage change. That's all it is. Just kind of rolling like you're doing the wave at a stadium from one part of the axonal membrane to another until it reaches the terminal point here. And then once it reaches this, it trips open all those calcium gates that releases these synaptic vesicles, and they release their acetylcholine, which is your neurotransmitter. And that neurotransmitter will open up a ligand-gated sodium channel, which will then create another influx of sodium, creating voltage change, which will then start to trip these voltage-gated sodium channels, and they'll start to propagate the action potential. And that propagating action potential will go down to the T-tubule, where it'll enter into the deep inner bowels of the muscle fiber. Wow.
Is there a good reason why there is a gap as opposed to like direct contact or direct contact?
That's a good question. So why not direct contact? Well, first of all, it's a little easier to rewire if there's not direct contact. The other thing is also, if there's like direct contact, electricity generally doesn't work well with direct contact in this particular case, because part of the secret sauce of this, the way you get the electrical signal from the neuron to jump over to the cell, is partly inherently a part of the fact that you have a disparity of charge separated by an insulator. In this case, the gap is acting like an insulator. So once the charge, so basically what would happen is it would just constantly happen. Like, just like if you were to walk up to a doorknob after shuffling along the carpet, you can't control that. The second you come too close, zap, right? You can't control it. You can't modulate it. You can't direct it. It just happens. And it happens however it wants. That's what happened if there's direct contact. By having the gap, you ensure that that won't happen. So even if that electrical signal coming through the neuron arrives right there, just staring at the muscle fiber, it's never going to do anything to the muscle fiber, because the gap is acting like an insulator, keeping it from arcing. So what that means is then you can control the nature, the quantity, and the behavior of the actual transmission by how much acetylcholine you produce. So the neurotransmitters are actually the way that your nervous system wraps its control freakish self around an uncontrollable quantity, which is the electrical nature of things. Electricity and electrical flow is random and chaotic, right? And so this is the cell's attempt to sort of take some of the energy, but to focus that and channel it in a controllable way. And the only way they can do that is if they sort of transfer that into a molecule that they can control.
So what starts the balance, or is it the acetylcholine and interaction with the calcium or the salt?
For the muscle fiber, it's the acetylcholine.
So that's basically the spark that starts the motion.
Exactly, right? So what happens is, as soon as acetylcholine binds to its ligand gate, what it does, it throws open that sodium channel and then inrushes sodium. Well, here's the problem. Sodium is an ion, which means it's packing positive charge. So anytime that you have positive charge, that basically means it's electricity, right? That's where you get the voltage effects of the nervous impulse.
From sodium, not the calcium.
But calcium also has a charge. It's also a 2+ charge. So it can have a very similar type of an electrical effect. But it's different, right? It's because it's a different ion. It's got a different power to it. So it's not quite the same. It's like a battery, right?
So
Some
the
batteries will just
acetylcholine,
basically
what is
light you
that?
up.
Is that like that acidic, or what is it?
What, acetylcholine?
Yeah.
It would be basic in nature. It's not acidic. But there's something that I want to do, because really there's no way to really understand this without doing it. It's going to take me a little while to sort of flesh this out. It's a piece of general biology. Here we go. but now's the time to do it because we're starting to talk about it. And it is kind of necessary to be able to wrap your head around it. And it's usually in general biology, we're talking about it during the cell transport section of the system. And I refer to it as channelology. So remember, a channel is a type of passive transport, right? And in passive transport, the one thing that we know is that the reason why it's passive is because passive transport is essentially the flow of molecules from an area of high concentration to an area of low concentration, right? That's true for all of them. If it's diffusion, it's a solute molecule going from high to low. Just think about putting like a drop of blood in water, right? The reason why it diffuses out is because the high concentration of the blood drop and the low concentration of the water are at odds with each other. So what happens is it goes from high concentration and spreads out to low concentration until what? Until it's completely equilibrium, right? In which case there is no more flow, right? So that's diffusion. Osmosis, which is the movement of water or the diffusion of water, is kind of also doing a similar thing. Well, in this case, what water does is it chases salt. As a matter of fact, in biology, we always learn osmosis by two words. It's the easiest way to learn osmosis. Those two words are salt sucks. Gotta say it with attitude. Right? Salt sucks. Just think about throwing up the double middle finger. You're like, okay, that's a little angrier than I prefer to be on a Thursday. Some of you guys are like, no. Yeah, you could be like, you could look right at me and say, salt sucks. Yeah, like a little passive aggressive thing, right? My kids are really good at that. It's like, do you want to tell me something? It's like, no, sucks. And it's like, okay, just go play somewhere. I know I'm not popular, just go play. So there's a couple of ways you can do this. And the reason is because we see this all throughout physiology in particular. And when it comes to especially things like osmosis and diffusion, you'll do yourself an abundantly good favor if you really get comfortable with it now, because I can guarantee you when you hit, especially the urinary system, osmosis and diffusion is going to come for you, looking for your head. Right. So if you're not comfortable with osmosis and diffusion, then it will devour you. Okay. So because a lot of these basic biological principles that we learn about in general bio really come out in physiology and AMP2. So that's the reason why I'm okay with kind of covering all of these here, because you're going to see manifestations of these throughout physiology when you get deeper and deeper into physiology. And that's kind of where we are right now. We're kind of getting more and more physiological, less and less anatomical and structural. So the first method, or the first way to kind of control flow through a channel, is basically with using osmotic control. So in this particular case, what you have is a cell. I'm just going to draw it. So here's my cell membrane, and I've got like a little protein channel. Looks like that. And in this case, I've got the outside, this is the inside, and here's what I'm going to do. I'm going to put a high sodium concentration on the outside and a low sodium concentration on the inside. So if this guy right here was a sodium channel, which direction do you think sodium would move? In, right? Because it's a solute, so it'd go from high to low. That means the sodium would go from like that. However, what about water? Like if this was a water channel, where would it go? Yeah, right, because salt sucks. So the salty area is outside, so water would naturally go outside, right? This is how we move water around the body, through channels, open channels called aquaporins. The way we control the movement of water in the body is not by telling water where to go. but by manipulating our salt piles to lure water in the direction that we want it to go. If I want water to go over here, what I do is I build up a bunch of sodium or salt over here, and naturally water is going to chase it. As long as I leave all those channels open so it can just freely pass through. Okay. So basically what you're doing is you're controlling movement through these channels simply by manipulating the osmotic environment, the concentration gradient. The second one we've already talked about is what's called a ligand-gated channel system. So in this case, I'm going to have the same system. I'm going to have high sodium on the outside, low sodium on the inside. Now what I'm going to have is I'm going to have a gate on my channels, and there's going to be a little pocket right here on that gate. That little pocket, an active site, is designed to bind to a molecule, usually a signal molecule, oftentimes a hormone or something of that nature. And what will happen is this particular molecule, when it binds to my gate, will change the shape of the gate and open that gate up. And so when I open that gate up, which direction is sodium going to move? Well, the high concentration is outside, right? So that means the sodium is going to rush inward, right? Because sodium is a solute, and solutes always move from high to low, right? That's basic diffusion. Okay, so that's a ligand gate. Now, what's the benefit of this one? Well, the benefit of this one is basically you can control movement by controlling this ligand. So you can make this molecule whenever you want, under whatever circumstances you want. And you can control whether or not you open that gate. Okay, so what is the next one? The next one is the voltage gated channels. We also talked about this one in this slide. And the voltage gated is a little bit different, right? So I'm going to start it off, by the way, with a ligand gate. I'm going to draw the same system. So I'm going to start it off with a ligand gate. And then I'm going to go voltage on you. So I'm a high sodium on the outside, my low sodium on the inside. Now, let's imagine that what we're going to do is we're going to basically bind to this gate first. I'm going to open up that ligand gate, right? And then what happens to sodium? Where does it go? It's going to rush in. Now, here's the deal. With a voltage gate, remember, it's not just sodium that's rushing in. Remember, sodium is an ion. So when you're basically bringing in sodium, you're also bringing in positive charge. Think about this for a second. Since sodium is an ion, when you have a concentration gradient, it doesn't just mean you have a lot of sodium outside. You also have a lot of positive charge. So as a result, the outside looks relatively positive because of all that sodium out there. The inside, relatively looking, looks negative. This creates a disparity of charge separated by an insulator, which creates what? A membrane potential, which is basically a fancy word for voltage, which is actually measurable in skeletal muscles. It comes in right around minus 70 millivolts. So think about it then. When you open up this ligand gate, in rushes this positive charge. What effect is that going to have on this membrane? Yeah, right? So you have all this positive charge rushing in. Then all of a sudden, what's going to happen is it's going to flip this charge, isn't it? That's a process called depolarization. It's going to make the inside now look positive because that's where all the positive charges rushed in. The outside now is going to look negative. That voltage change is sensed by the voltage gates. When it senses that voltage change, it pops open, and then what happens? In rushes more sodium or more positive charge. And what does that do? It's going to depolarize the next piece of membrane, which is going to be sensed by the voltage gate, which is going to pop that open, and then in rushes which is more positive charge, which is then going to flip the charge again, which is going to be sensed by that last voltage gate, which is going to inrush. It's like a series of dominoes, one tipping the other over in what is called a rolling depolarization. That rolling depolarization is what we refer to as action potential. That is the electrical wave that is rippling down the axon that arrives at the presynaptic membrane that triggers the calcium channels, which are hanging out there, to release, because those are also voltage gated. And when they release the calcium, that tells the vesicles full of acetylcholine to dump its acetylcholine into the synaptic cleft. And what's waiting across the synapse? Ligand gated sodium channels. And who's their ligand? Acetylcholine. Right? So the acetylcholine will come in, bind to, and that's exactly what's happening here, right? The acetylcholine will come in to the muscle fiber side, bind to that acetylcholine gated channel, opening it up and reigniting this new round of dominoes tipping over. And that is basically what's rippling through the sarcolemma and what dives down into the T-tubules where it hits the triad. And then that rolling voltage change in the triad triggers another set of voltage gated calcium channels. Only in this case, they're in the sarcoplasmic reticulum, who we know is storing what? Calcium. So once those gates are triggered because of that nosedive of action potential coming down the T-tubule, it'll open up the sarcoplasmic reticulum channels, outrushes all the sarcoplasmic reticulum calcium, bathing your myofibrils in calcium, going for who? Who binds the calcium? We just learned about them. Troponin, right? The calcium will bind to the troponin. Troponin pulls the tropomyosin off of the G-actin sites, allowing the myosin heads to pull, and then you get sarcomeric shortening. Wow. There's one more that I want to cover here because it's in our sensory system.
Yeah, like every time you
Milliseconds. Yeah. Yeah, milliseconds. It's very fast. It's like a flash. So the other one is a mechanical gated. This one you see actually in your inner ear. This is actually kind of cool. So in this particular case, what you have is a gated system. There's your gate. Only now you have kind of like a hinge protein that's attached to it, that's then attached to a distortable structure, a protein or something like that. So if I distort this protein, I'm basically going to fold it over this direction. What's going to happen is it's going to pull on this hinge joint and it's going to pop open that little gate and rushes your sodium. This is actually how you hear, because your distortable structure in your inner ear is a membrane that's tethered to a hinge protein that's tethered to the gate on your sodium channels in your inner ear. So what happens is when the airwaves of sound, like the way you're hearing my voice now, it's compressing the airwaves. And so the waves that are air coming at you, it has compression waves to it, right? It has amplitude and wavelength to it, depending on the pitch. Like if it's really high or it's really low, it's got a certain lollop to it or a certain sharpness to it. And so as it comes to your external acoustic meatus, lingo, right? It's going to hit your tympanic membrane, lingo, right? And it's going to basically vibrate your eardrum. And then the ossicles, the auditory ossicles, right? The hammer, the stirrups, all that stuff. Those are going to basically vibrate. And on the other side, they're attached to a thin little membrane that on the opposite side of that is connected to the inner ear fluid, the endolymph of your inner ear. So what happens is ultimately the airwaves hit your eardrum, vibrating your eardrum, which pull on those auditory ossicles, and those pull on the membrane like a wave machine, right? Which is constantly going back and forth, creating waves like that. And it's creating waves that are exactly identical to the airwaves that are hitting your membrane. So it's doing this. And so you're recapitulating those waves in the endolymph in your inner ear. And as those waves wash over this distortable membrane in your ear, some of those waves will be slight and gentle, and they'll distort the membrane a little bit. which will open up the gate just a little bit. And in will rush just a little bit of sodium. You hear that as a certain pitch. Some of those waves will be very aggressive and they'll do a lot of distortion and it'll really open up the gate, and in will rush a lot of sodium. You hear that as a different pitch. So your ability to hear pitch is associated with this mechanical gating system. So there's a lot of reasons you could go deaf, but one of them is potentially that. Like if you physically max out or compromise the ability to distort that membrane, or if it becomes undistortable, then you're not going to be able to hear pitch at all. Yeah, that happens all the time. My wife gets it all the time. She can't go over the mountains. I always tell her, it's like you're living in like the worst state of the planet to have ear issues. But no, the membrane's not bad. I mean, they can always patch up the eardrum. That's not a big deal. But basically what's happening is you need a complete eardrum in order to be able to kind of maximize the waves, to sort of be able to transmit the waves. If you've got a big old hole in there, which is what happens when you blow that out, usually due to pressure, oftentimes because you have an inability to clear out the fluid backflow in your eustachian tubes. So that's a lot of what it is. And so you get pressure buildup in your inner ear, and then it just blows your drums, especially if you start to add elevation to it, and it kind of just, you have no way to release it. That's the reason why our ears pop. We're kind of like feeling the calibration of the eustachian tubes kind of recalibrating itself and trying to keep those internal pressures calibrated. But if somebody has a blocked eustachian tube, then it just builds up and builds up and builds up, and they don't have a way to release it, and then eventually it's going to go out the eardrums. When that happens, you just put a big old hole in your eardrum, and you're not going to be able to hear because you can't vibrate your eardrum anymore. But eventually it'll heal itself up, or if you go to a good ENT, they can patch it up pretty well. So it's not necessarily a catastrophic thing. You can get it back. This is the reason why a lot of times people have tubes in their ears, is to kind of allow the release of pressure without compromising the eardrums. But yeah, I mean, that's kind of part of it. But I mean, that's not necessarily a permanent thing. So I mean, it's just painful. It doesn't feel, yeah, it doesn't feel good. But eventually you'll be able to hear again. And it's just like putting, like if you're in a drum, like if you got a drum at home, and it's like you have to have a tight membrane in order to get the sound. If you just put a hole in then you're not able to get a sound out of it for the exact same reason. So, okay. So what we'll do is we'll pick up with the sliding filament model on Tuesday, and then we'll probably the end of Tuesday will be the exam cutoff. So wherever we end at that point. So. Yes.
So will that bottle or the drinks